Hello and welcome to another episode of Microbially Speaking. I'm your host, Kevin Stinson. If this is your first time joining us, then welcome to the channel. If you're a returning listener, then welcome back. So today, as promised, we are taking a step away from Ebola, away from all of these fun special viral pathogens, and talk about something a little bit more positive. Just a few weeks ago in early June, the lab of Dr. Jerry Wright at McMaster University in Hamilton, Ontario, published an article in the journal Nature about a newly discovered antibiotic candidate molecule. Now, it's likely no surprise to any listeners of this channel that we are currently in the midst of an antimicrobial resistant crisis. For decades, our ability to discover novel antimicrobial drugs and then bring those drugs to market has been far outstripped by bacteria's ability to develop and spread resistance mechanisms. It's truly a tale as old as antibiotics themselves. And truly, ever since humanity first began harnessing antibiotics as a modern day therapeutic, we've seen issues and impacts of antimicrobial resistance. Even some of the very early day antibiotics often had resistance identified in clinical isolates before the drug was even widely available on the market. Now, for bacteria, antibiotics are just a tool. They're a tool that's developed over billions of years with the purpose of trying to kill competing microbes. It wants to reduce competition in a natural environment so that it can reduce the amount of demand and competing organisms trying to take its own nutrients, resources, or the habitat itself. And likewise, other bacteria then develop biological countermeasures to ensure their own survival. And that becomes resistance mechanisms. Now, when we look at a lot of early day antimicrobials, antibiotics, these were just simply finding these molecules in the environment, purifying them, and using them in humans. And so resistance was already well established in nature. Add to that decades of overuse and misuse of antibiotics, and simply running out of the easy to discover antibiotic compounds in nature, things that are really easy to find, purify, and exploit. And we're left with the reality that we see today in that we are running out of antibiotics. We're rapidly approaching an era where we may see an end to antibiotics as an effective treatment for many complex infections. And so the pressure's on to find novel drugs to continue this fight against infectious disease. Now, the story of discovering antibiotics, antimicrobial resistance, all of the history that goes on there is absolutely fascinating. It spans 80 years of innovation and science. But let's save that for another episode. And instead today, let's focus on what we're discovering with these new drug candidates. McMaster's been in the spotlight a few times in the past couple of years for their work on these novel antibiotics, or rather novel antibiotic candidate drugs. And I'll go back to that term of these are candidate drugs, not full-fledged antibiotics yet. For today, we're going to look at the three most recent or the three most well-publicized versions or three most well-publicized candidate drugs that are coming out of McMaster over the past couple of years. We're going to look at what these drugs are, how they work, how they might then be developed into clinical use, and some of the hurdles and roadblocks we might see between now and hopefully getting them to market. Now, the three drugs we're going to be following, there's two from the lab of Dr. Jerry Wright and one from the lab of Dr. Jonathan Stokes at McMaster. The drugs are known as enterololin, lariocidin, and the most recent one, the one that just came out a few weeks ago, manikomycin. Now, it's always fun to look at these complex names and think through, like, how did those names come into being? And I don't think I'd have the creativity needed if I was ever to discover a new drug or a new organism to figure out some kind of creative naming scheme for that organism. So let's start with enterololin, the drug discovered, or the drug candidate rather, discovered in the lab of Jonathan Stokes. So the name enterololin likely refers to a combination of its spectrum of activity and its mechanism of action. Admittedly, though, I might be speculating a wee bit here. So enterololin has a really unique narrow spectrum of activity. It specifically targets the family, the bacterial family called the Enterobacteriaceae. Now, these are often just referred to as enterics. And many of these bugs are found in the gastrointestinal tract of humans and animals, and many of them are also medically important. Medically important meaning that they routinely cause disease in humans. Now, this includes some of the most common bacterial infections causing gastrointestinal illness, and things that often hit the media, often hit headlines, and are names that even lay people will likely recognize. Things like E. coli, Shigella, and Salmonella. Things that cause, or bacteria that cause, gastrointestinal illness through contaminated food or contaminated water. Now, this is where enterololin has a pretty cool advantage. So despite being called the enteric bacteria, these Enterobacteriaceae actually only make up a really small percent of the gut microbiome, less than 1%. And if you could recall back to our very first episode, the gut microbiome, the bacteria that naturally live and reside in our gastrointestinal tract, is made up of somewhere around 40 trillion bacterial cells. And most of those are important. Most of those are good bacteria. We want them in our GI tract. And so this is a drug that is very unique in targeting just a very small amount of the bacteria that can sometimes cause disease without hitting a large number of the bacteria that are the healthy, normal flora that we want to keep there. So to this end, enterololin has is referred to as a microbiome sparing antimicrobial, meaning it can just hit those bugs without hitting everything else. Now, the research behind enterololin also focuses on its use primarily in things like irritable bowel syndrome and Crohn's disease, where we often see that flare-ups and illness is caused by dysbiosis, by an irregular ratio of these Enterobacteriaceae compared with other normal flora. And so this is a really interesting drug in that it can help to reestablish some semblance of symbiosis, some semblance of balance in that gut microflora by targeting just those Enterobacteriaceae and sparing the rest of our flora. So it'll be really interesting to see what happens with this drug as it goes through development. Anyway, that's where we got the entero side of the name. But what about the second half, the lolin? This probably refers to the target of the drug. It's a component of the bacterial lipoprotein transport system. And I love this name. It's called the LOL protein, or specifically a machine called LOL-CDE. What LOL-CDE does is it's a transporter. It's used to take proteins that are embedded in the inner cell membrane, the inner plasma membrane of a bacterial cell, and move it to the outer membrane of a bacterial cell, specifically in these Enterobacteriaceae. Now, without this action, without LOL-CDE, structural proteins can't get embedded in that outer cell membrane. The cell membrane destabilizes and the cell dies. And so enterololin acts to block that function and, in essence, kill the bacteria. So we got Enterobacteriaceae, lolin, it targets that LOL protein complex. Now, the next of these three drugs that we're looking at today is called lariocidin. And again, we can look at the name. Lario, that prefix, comes from the name lariat, which I had to Google, and is a technically more technically appropriate name for a lasso. It's also like the name of a weird kind of tie where it's like a rope and a metal piece there. Someone is going to listen to this going, I should definitely know what a lariat is. as a tie. But either way, lariocidin is referred to as, or is what's known as, a lasso peptide. So what do I mean by this? It is a small peptide, a small string of proteins that on one end you have a ring and it has a long tail. And during the process of translation, of protein synthesis, that tail gets looped back through that ring, and you get this folded knot-like protein, or a lasso, or a lariat. Now, this gives the peptide a fair bit of advantage in stability. It becomes more heat stable, it becomes more resistant to degradation, stable in the environment. And if we think back to how our bacteria producing antibiotics and why are bacteria producing antibiotics, it's often to release it into the environment to kill competing organisms. And so having something that's highly stable in that environment makes a lot of sense from an evolutionary standpoint. Now, lariocidin acts to inhibit bacterial ribosomes. And this is the spot where you have bacterial mRNA getting translated or synthesized into proteins. And so lariocidin hits that ribosome and halts protein translation, protein synthesis, or otherwise induces miscoding, misfolding, and creates non-functioning proteins. This gives us the suffix "cidin" in lariocidin, referring to a compound which inhibits or kills an organism or inhibits growth. It acts to actually kill the bacteria. So lariocidin, a lasso protein that kills bacteria. Now, one of the coolest parts of this whole lariocidin story is how researchers found it. Because literally, researchers went to their backyard garden, scooped out a bunch of soil, and said, let's see what we can culture out of this soil. Now, that's not a novel idea. A lot of soil bacteria produce antibiotics that we use every day. What's unique here, and the challenge that the Wright Lab had to overcome, is that bugs that grow in soil are often really hard to cultivate in a lab. And so the Wright Lab looked at ways of developing very, very slow growth mechanisms to purify out some of these really, really hard to come by organisms, things that would have been overlooked in previous studies. They were able to take their backyard soil experiment and culture out bacteria over the span of a year. So a year to grow some of these slowest growing samples. One of the bugs they pulled out was a bacteria by the genus Bacillus, and it produced the peptide known as lariocidin, or rather that they named lariocidin. Now, unlike enterololin, unlike that first drug candidate we talked about, lariocidin is a pretty broad spectrum antibiotic. It's very, very similar to a lot of other human antibiotics that we see. It hits a ton of human pathogens, but it also would have widespread impacts on gut microbiome and other normal flora. It's also not by any way, shape, or form the only antibiotic that targets ribosomes. Ribosomes are a target that tons of antibiotic classes exploit to try to kill bacterial cells. What is unique about it, though, is it's hitting a previously unused target site. It's hitting a section of the ribosome that no other antibiotic that's in use actually targets. And this is unique and important because what it means is this can become a scaffold to develop other semi-synthetic and synthetic antibiotics off of this drug in future. Now, so far, both enterololin and lariocidin, we've talked about them, we've talked about where they came from. Let's turn to our most recent discovery, the manikomycin. And this one has a bit more of a unique naming convention to it. So lariocidin, enterololin, both of those had really scientifically, logically derived names. Manikomycin has a little bit more of a touch of creative flair to it, if you will. So we'll start with the second part. Mycin simply refers to the bacteria it was isolated from, Streptomyces. Streptomyces rhimosus, to be exact. And so manikomycin, that's where we get the mycin section. But the prefix, the manic part, is where it gets really interesting. This is where the creativity comes from, and this is straight out of the publication itself. So manic reportedly refers to the Hindi word meaning ruby or precious gem, and it's meant to reflect the significance and the rarity of finding a novel antibiotic and an antibiotic with really its own unique mechanism of action. As an aside, I don't speak Hindi, so I relied on Google Translate to confirm this, and it seemed to be accurate. So you'll have to let me know in the comments if you speak Hindi, if this was actually an appropriate translation. So similar to lariocidin, manikomycin acts directly on bacterial ribosomes, and it interferes with that process of protein synthesis or translation. It also has its own unique spot that it binds to the ribosome that previously has been unexploited and unutilized. So it's somewhat unique in how it hits the ribosome. What is unique here is that both of these drugs seem to kill the bacteria. Most of the antibiotics that hit ribosomes are what we call bacteriostatic, meaning they slow bacterial growth, but they don't actually kill it. It relies on the immune system to come in and kill the bacteria. These two drugs, lariocidin and manikomycin, are at least appear to be bactericidal, meaning they will actually kill the bacteria. And that's really important. Bactericidal activity is usually preferred, especially in severe infections, as it becomes a little bit easier to dose and maintain effective treatment strategies. Now, I've perhaps been a bit overgenerous rather, to McMaster so far, at least in suggesting that they're the only people discovering novel antibiotics or antibiotic candidates. The reality is, in the past 5 years, we've seen at least 10 other drug candidates identified by various groups across over the world. Some of these are hitting the same targets that we've talked about. So there's been two other candidates that hit that same LOL protein that enterololin targets. We have lolamycin, which has a very similar spectrum of activity against Enterobacteriaceae. And we have abaucin, which is a little bit more narrow. It specifically seems to hit a bacteria called Acinetobacter baumannii. So there are other groups out there. There are other people finding some of these same drugs or very similar drugs. Although, to be fair, that last one, that abaucin, was actually discovered by the Stokes lab, the same people who also discovered enterololin. So another McMaster success story there. The big takeaway here is we are not at the end of a drug discovery era. That era is not over. We continue to find new potential antibiotics. We're finding new targets of things that these antibiotics could hit. And we're even discovering what could be new classes of antibiotics. Some of these drugs have even started to show potential inhibition against some of the most resistant bacteria. The ones where we're closest to running out of antibiotics, we're finding candidates that can hit those bugs. And so does it mean that the antimicrobial resistance crisis is finally nearing its end? No, not by a long shot. Unfortunately, at least two of the candidates we talked about today, lariocidin and manikomycin, are naturally occurring molecules. They are found directly in nature, and that means that there's already been selective pressure to develop resistance mechanisms. It would then only be a matter of time for that resistance mechanism to make its way into clinically relevant organisms and then start disseminating. Even enterololin, which is a fully synthetic molecule, not naturally occurring, history would suggest that it too would only be a matter of time before we see resistance to it start to emerge in clinically relevant organisms. But like we said earlier, some of these represent entirely novel drug targets. And so that creates a potential for some long-term innovation. I used the term a scaffold previously. They'll create a scaffold for future drug development. And what I mean by that is we could take the core of the drug and research could start to look at how can we manipulate that drug chemically to create semi-synthetic analogs. Now, these synthetically developed and derived analogs, you can do things like further refine what will it target, the spectrum of activity, where it traffics through the body, or develop analogs that are more resistant to resistance, or rather escape resistance mechanisms. Well, for a second, consider penicillin. Penicillin is a natural product. It's been decades since it was discovered. And in that time, there have been a huge number of semi-synthetic analogs that have come onto the market based on penicillin. And there are a lot of drugs that we still use widely today, things like ampicillin, amoxicillin, cloxacillin, piperacillin. Each of these are small little tweaks and changes that subtly alter how the drug interacts in our body, what bacteria it targets, how it can be used clinically. And so that's the potential we have with some of these novel antibiotic candidate drugs, is that they'll create this whole scaffold for future rounds of drug development. So now, when can we then expect to see these drugs hitting the market? And unfortunately, that's where it's really not an easy question. As exciting as these drug discoveries are, there's no actual guarantee that they'll ever become drugs ready for human use. And so to unpack why, we have to look at this from a few different lenses. First, we need to appreciate how different laboratory-based testing is, or in vitro testing, in a lab testing, from actually seeing how does this drug interact and work in vivo, or in a person. So broadly speaking, we can think of antibiotics as needing a minimum amount of drug, or minimum concentration of drug, in order to kill a bacteria. And we call this the minimum inhibitory concentration, or MIC. In a lab setting, it's super easy to test this. You take your liquid media, you add in your bacteria, and then you add in different concentrations of the antibiotic until it stops growing. It's really easy to control how much antibiotic is getting into that tube. But it gets a little bit more complicated when things hit the body. We have a ton of different factors to consider, like how does that drug actually move and get processed through the body? And so we have two terms in pharmacology to look at this. We have something called the pharmacodynamics and the pharmacokinetics. We'll go with pharmacokinetics first. Pharmacokinetics looks at how is the drug absorbed, distributed, metabolized, and excreted from the body. So for example, if a drug is needed if a drug needs to be in the bloodstream in order to target an infection, but can't be absorbed across the gastrointestinal tract, then you can't deliver it orally and expect it to do any good work. And that's exactly what we see with vancomycin. Vancomycin, really, really potent drug used for resistant staph infections, or MRSA or MRSA infections. But if that infection is in the blood or in the tissue, you need to get the drug there. And vancomycin is not bioavailable orally. You can't take it orally and expect it to hit systemic circulation. And so you have to give it by an injection. And so how that drug gets into the body has a huge impact on how it will be dosed. Once in the body, it starts to circulate, it starts to be distributed, and different drugs will accumulate in different tissues at different amounts. Some tissues will tend to concentrate the drug, other tissues will completely get missed by the drug. If you have an antibiotic that you need to treat meningitis in the brain, but this antibiotic won't cross the blood-brain barrier, it doesn't matter if the bacteria is susceptible in a lab setting, it can't be used there clinically. But then you have to look at how does the body metabolize or excrete the drug. So how does it metabolize it to alter it, break it down, change the drug, and then how does it purge it from the body? All of this means that the drug will accumulate in different tissues at different rates, at different concentrations. And so that then becomes really important to take into consideration when you think of how would I dose this drug in order to get it into appropriate concentrations at appropriate sites. And that's where we tie it to the pharmacodynamics, the other half of this equation. Because pharmacodynamics looks at how does the drug actually function. This is where we get into that concept of the minimum inhibitory concentration. How much antibiotic do I need to kill my target organisms? And is that changed by the specific type or area of the body that I'm working in? Does it change when I'm looking at how does it interact with other aspects of the body? Does that MIC increase or decrease? Now, given all of these concepts, we then have to ask the question, can I reasonably hit a minimum inhibitory concentration in the tissue I need it to with the drug in a reasonable dosing criteria? And if all that checks out, we still have to then ask the question, is the drug just going to kill the patient? Because if the drug kills the patient, it's not a very useful antibiotic. And so finding a novel antibiotic target, huge milestone. But it then starts a very long, very complex, and invariably expensive process of validating that everything we could see in a lab works in a living organism. So that would include things like animal studies and animal models to see how does it move through a body, where does it accumulate, how does it work in vivo or inside a living animal. It includes toxicity studies, initially looking at human cell culture to make sure it doesn't kill human cells, and then eventually in humans themselves to make sure it doesn't impact us. And only then does it enter into multi-phase clinical trials to confirm, does the drug actually work? Is it actually effective as an antibiotic, or is it just something that works well in a lab but not in a clinical setting? All told, these steps can take 10 or years to a decade and cost hundreds of millions of dollars. But let's say we have a novel drug. We know that it works in a lab. We've done animal studies. The animal studies show that the pharmacodynamics and the pharmacokinetics are favorable for its use. We know that it's then been shown to be effective as a therapeutic in humans. It doesn't kill us. Even with all of that, the drug then still has to go through a regulatory approval process, which that can take time. And then a company needs to be willing to market the product. And unfortunately, there's not a lot of money in antibiotics. So many recent approved marketed antibiotics on the market are very niche products. They're very narrow spectrum drugs, or they're drugs that are intended to only hit extremely resistant organisms so that they are, in essence, brought on as drugs of last choice. They're almost all, or they almost have all been synthetic analogs or reformulations of previous drugs, and so that gives it a very narrow functional use. They're extremely expensive to bring to market. They're extremely expensive to develop and get through testing and approvals, and so that means they come with a huge price tag when they're finally available for use. And if the market's not big enough, it might not actually be worth it for a company to say, you know what, I'm going to take this drug, go through the approvals, and bring it to market in a given country. And that is unfortunately the reality in Canada. In 2020, there was a paper published that looked at access to 18 recent antimicrobials, so antimicrobials brought to market between 2010 and 2019. And it looked at 14 high-income countries to see where were these drugs accessible. Somewhat unsurprising, US led the pack here. They had access to 17 of these drugs. Japan and many European countries all had access to between 5 and 10 of the drugs. At the time of its publication in 2020, Canada's access was 2 drugs. Canada simply is not that big of a market, and so it's incredibly difficult for these highly niche, targeted, narrow spectrum or narrow clinical utility drugs to be brought into a very small market, given the cost of the drugs. And when we think about it, that barrier becomes significantly higher when we look at middle and low income countries. Canada's a high income country and it shouldn't be that hard to bring it in, but low income countries that bear the brunt of antimicrobial resistance challenges have even more issues and difficulties bringing them to market. Now in 2026, figures are a little bit better off. Of those 18 drugs, we are now up to, I believe, 6 on my last check. 6 currently in market, and maybe 1 or 2 others approved but not yet marketed. But that means we still have a ways to go. And since 2020, other drugs continue to get onto the market in other countries and haven't hit Canada at the same rate. So we said, though, we're going to focus on the positive today. And so let's take a moment to just celebrate and acknowledge the work that's being done at McMaster in the Wright lab and the Stokes lab. They're finding new drugs, new drug candidates, and they are making good forward momentum into creating that sense of that safety net against antimicrobial resistance. There's tons of hurdles that they have to go through. It's going to be years before they will eventually get them into the market if they can, but it is absolutely an important step forward in that fight against antimicrobial resistance. And it's working to stave off an era where we might have to see antibiotics have failed. Anyway, let's leave it there for today. If you liked today's episode, be sure to give it a like and subscribe to the channel or follow on Spotify. If you have any questions, any comments, anything you want to hear more about on this topic, be sure to leave a comment below. If you have any topics you want to see on future episodes, let me know in the comments and we'll see what we can work in. I hope you enjoyed the session today. Thanks so much for listening or watching today, and we'll see you next time on Microbially Speaking. Bye now.